Yeah, good evening, friends. Good evening, friends. This is Chandrasekhar. Okay, today we will see some interesting concept that is a vaccination. Let's understand something about this vaccination little briefly. First of all, friends, what exactly the word vaccination means and understand the concept why we will use the vaccines basically. Friends, we know that vaccination basically a process. What kind of process it is? It is a process of giving just 1 second, friends. Yes, yeah, vaccination is a process of giving antigen. Why we will give the antigen, my friends? We will give the antigen to stimulate the immune response through active immunization process. So, my friends, actually, in the form of vaccine, we will introduce the antigen. Once we introduce the antigen, our immune system will get activated. The moment our immune system get activated, it immediately start showing the response in the form of production of the antibodies, is it? And friends, also understand very briefly one important point, that is there are different type of vaccines are there, friends, but our ultimate goal of the different type of vaccines are injecting the antigen, is it? But friends, we never ever inject the pathogen. That you have to remember for the exam without fail. Why I am saying that we are injecting the antigen? Of course we are injecting the antigen to stimulate the production of the antibodies. We are not at all injecting any pathogen. We are not at all initiating any disease, my friends. What we are injecting is just a antigen. Only we will stimulate the antibody production by injecting the antigen. We are not at all stimulating any type of the pathogenesis. So please remember that without fail, friends. Right. So understand once again very briefly. See here, there is a small survey. If you see the picture very briefly, my friends, whatever you are seeing in this particular blue color, yellow color and red color. Blue color are the people, they are not immunized. They are not immunized, my friends. Even though they are not immunized, they are healthy. Okay, then the next one, this yellow color people, what you understand, they are immunized. They are immunized. Whereas this red color people, they are sick and they are contagious. Okay, and if you see, my friends, in this particular entire blue color population, 99% of all the people, 9% of all the people, my friends, they are completely healthy. Even though they are not immunized, they are completely healthy. Just 1% or 0.5% of the population got affected. But my friends, it's more than sufficient to convert this entire healthy population into a contagious population. We will call na pandemic. You take an example of the coronavirus. Correct, right? Even though 2 to 3 people got affected, if they are in close contact with the community of the 100 people, the disease can easily spread very, very, very fastly. And second and most important thing, so if you see this picture, my friends, in this particular population, there are certain people are there who are not immunized, that is blue color. There are certain people are there who are immunized, and certain red color people are there which are sick and which are contagious. So my friends, once again, this red color, that is who are sick and contagious, they can spread the disease through some of the population. But my friends who are already immunized, they are being protected over here. And some blue color people, they are not immunized, but they didn't get diseased. Why? Because of their stronger immune system. right? And in the third condition, if you see, most of the people are what? They are immunized, and very few are not immunized, but they are healthy, and very few people are diseased and contagious. My friends, even the very few people are diseased or contagious, they are unable to spread the infection. Why? Because my friends, because most of the people are surrounded by the people who actually already taken the I mean to say vaccine. Because of that, the sick or contagious people, they are unable to spread this particular infection. So actually, Vinayak, I'm coming to that point. What does it mean by the word herd immunity? Herd means, Vinayak, we call group. We call it as what? Group immunity. Because Vinayak, whenever we are giving an antibody, right, whenever we are giving a vaccine, we never consider for a single people or 10 people or 100 people. We will always consider here a group of people. We will always consider a multiple people. Herd in the sense a group or a community. Okay, so the vaccine which was administered to the group of the people and group of the community, that's what we basically call it as herd immunity. Okay, and there are different types of the vaccines are there. What are these different type of vaccines, friends? See, there are attenuated vaccines are there, then inactivated vaccines are there, as well as toxides are there. See, attenuated live vaccine. What does it mean that, my friends? We are directly giving the antigen. So understand, very, I mean to say that directly we are using the microorganism, but we will attenuate it in the sense we will use that microorganism which is in the weakening condition. So attenuated vaccines we basically called as a weakening vaccine. Inactivated means it's a whole organism. Whereas toxide means, my friends, instead of taking the whole organism, we are going to take a certain part. So we can take their polysaccharide, we can take their polypeptide, we can take even their surface antigen in the form of a recombinant vaccines also. These are all the most important vaccines that are available at present. Of course, all vaccines shows the great result, but let's understand one by one vaccine a little differently, a little detail. First one, attenuated live vaccine. So, my friends, what exactly we are doing in the attenuated live vaccine? If you see the picture very briefly, what we are doing first, we are introducing the vaccine. We are introducing the vaccine that containing killed or weakened organism. Majorly, we will take the weakened organism. Now we have the introduced that particular antigen. Now that particular bacteria which is having the surface antigen, that particular surface antigen was recognized by our lymphocytes. Is it? Okay, so once it can recognized by our antigen, okay, once it get recognized by the antibody, I mean to say that B lymphocyte, our B lymphocytes are going to recognize this particular surface antigen. Now, my friends, this particular B lymphocytes, we will be calling it as a naive B lymphocytes, right? So because why we are calling naive B lymphocytes? Because without antigen exposure, my friends, they are not too great active. Upon the antigen exposure, upon the antigen exposure, the naive B lymphocyte completely get active. Once the naive B lymphocyte can completely get active, it undergoes division and it is going to produce the plasma cell, then followed by the memory cell. We know that plasma cell secrete the antibodies, as well as it is also secrete the, and memory B cell will secrete the memory antibody. This is what kind of response? Primary response. What exactly we did here? What exactly we did here? What we did is we have introduced the antigen in our body. Why we have introduced the antigen in our body? In order to stimulate our immune system to produce the antibodies against that particular pathogen, and at the same time we are storing the copy of that particular antibodies also whenever we exposed in future to the same kind of the pathogenic molecule. Now you understand. This is whatever we have did so far, my friends. It's a primary immune response. Is it? Now understand the concept. Secondary response. What is the speciality of the secondary response? What exactly you understand by the word secondary response, friends? Now really that particular pathogenic organism enter the body. Really that particular pathogenic organism enters inside the body. Once really that particular pathogenic organism enters, immediately what is going to happen, my friends? Now whatever the memory antibodies that are already stored in the body, friends, here once again we no need to show any kind of a primary response. Why? Because same kind of antigen we already injected in our body, we stimulated our immune system, we produced the antibodies also. Now what we are doing, my friends, now we are activating our secondary immune response whenever we exposure to the same kind of the pathogen. Is it? Then immediately what happen? Our memory cell will secrete that memory antibodies. Whatever the stored memory antibodies are there, it will directly will go and attaches to that particular surface antigen, and they will try to destroy this is what we called secondary response. So in the primary response, initially we have injected that particular antigen in our body, and we started stimulating our antibody production, and we have stored the copy of antibodies. Whenever we expose it to the same kind of infection, what is going to happen, my friends? Now immediately what is going to happen? So whatever the antibodies that we are stored, that comes into the picture and try to destroy that particular pathogen. Okay, that's what we call it as what, my friends? Attenuated live vaccine. Another one, my friends, whole organism vaccine. So this is also very important. Please remember the examples, friends. Live attenuated vaccine. So live attenuated vaccine means, my friends, we are taking whole vaccine here. Whole organism we are taking. Okay, so these all the organisms are basically grown in the abnormal culture condition. Friends, what does it mean by the word abnormal culture condition? Where initially they cannot survive for the longer time. So what we did, we have grown this all these particular microorganism in the abnormal culture. Once they have introduced in our body, they are unable to show any kind of the pathogenicity. Example, BCG for the Mycobacterium bovis. Now what this mycobacterium we did, we have grown this mycobacterium nearly 10 to 13 years in a particular type of the bile culture. Where we have grown, my friends, we have grown a particular type of the bile culture. My friends, whenever we have grown it in the bile culture, you know what basically happened? Whenever we have grown in the bile culture, my friends, understand, it is unable to survive whenever it enters in our body, because the bile conditions are different, and whenever it enters inside our body conditions are different. Hence, once it enters in our body after growing in the bile condition, it cannot survive for the longer time, and it cannot show any pathogenicity. And Sabin, this is the vaccine for what? For the polio, which was grown in the monkey kidney epithelial cell. Rubella virus, there are different stages are there rubella virus, but initially we have grown in the duck embryo cells, right? This is what entire whole organism. Now in the whole organism only, see, first of all, you have to understand how we are going to inactivate this particular bacterial vaccines. Friends, keep it in your mind. We are going to inactivate this bacterial vaccine either by heat treatment or by the chemical treatment also. So, heat treatment, what my friends? By increasing the temperature, we will destroy their biomolecules like protein, polysaccharide, their entire epitopes. We are going to, I mean to say, entire biomolecules we are going to be destroyed by increasing high temperature. Chemical treatment, we can cause majorly a physical damage by using the chemical treatment. And friends, this inactivated viral or bacterial vaccine, they never provide the cell mediated immunity, my friends. They will only provide the humoral immunity. Please remember that. Then polysaccharide vaccines. I mean to say that what, instead of using the whole organism vaccine, my friends, we are exclusively using the capsular polysaccharides. Only that particular area of the capsular polysaccharides only we are using. In this, we are activating the B cells in the thymus independent manner. We know no, the major antibody involved here is immunoglobulin M. You know na, immunoglobulin M is present on the which is expressed on the B cell first time. We will be calling it as a pentameric antibody. So only we are using a certain part of that particular bacteria. So, so that is capsular polysaccharide. You can say simply vaccine again is the Streptococcus pneumonia. Toxoid vaccines. What I told you, the exotoxins which are present in the bacterial body that are completely inactivated by a chemical treatment, that is especially by formaldehyde. As a result, whenever that particular toxoids enter in our body starts synthesizing antitoxide antibodies, and they will start degrading or destroying that particular toxoids. And most important one, here also epitopes should not be altered. Whatever the epitope initially we have introduced, same kind of the epitope we have to introduce. Example, tetanus toxoid as well as the diphtheria toxoids. And friends, there is another different type of the vaccine is there. That's what we basically called recombinant antigen vaccines. Okay, so actually, what is the specialty of this recombinant antigen vaccines? The genes which are encoding for any immunogenic protein. Any immunogenic protein, it can be cloned and it can be expressed in the bacteria as well as the yeast. Okay, and that will be used as what? Majorly vaccine. Suppose example, this is the hepatitis B virus. From this hepatitis B virus, by using different rDNA technologies, we have isolated the particular surface antigen. Once we have isolated the particular surface antigen, we are going to insert it into the vector where we have taken the I mean to say that with the help of a vector we are introducing into the yeast. Now, this particular isolated surface antigen gene, it will get modified in the yeast and it produced HBS, hepatitis B virus antigens. From this, we have isolated the vaccine and later we can use in order to destroy that particular antigen. Okay, this is what a major type of the hepatitis B vaccine. So these are all the different types of the vaccines. Okay, so friends, please kindly remember all the type of vaccines without fail. Once again, in the next class, in the tomorrow class, I'm going to discuss about the 100-day strategic preparation to crack the IIT Jam 2021. Please kindly attend that particular class and make use of it. Thank you.